Hello, good morning dear students. Hope all of you are doing well at home. I know it's not a relevant question at all. Then also, I know that we are all passing through a severe crisis. All of us are passing through this time. So let us fight against it. Let us stand united in this crisis. So let us pray for the better future of our nation as well as each one of us. So let us start. Are you ready for the revision? I know, I am sure that you are ready for the revision. So what we have learned so far, in the previous session what we have learned was regarding cell. What is the cell we have learned, the discoveries of the cells we have learned, the differences between unicellular and multicellular organisms we have learned, and the last we have learned was the differences between osmosis and diffusion, the differences between the hypotonic, hypotonic and isotonic. And the last point that which we have revised was that cell has three prominent features. What are they? The first one we have discussed, that was plasma membrane, and the second one is nucleus, and the third one is cytoplasm. The first one we have already learned. What is the plasma membrane? It is the outer covering of the cell organelle. Cell. So let us learn the second one about nucleus. So what is the nucleus? Nucleus means it's a prominent organelle that which is present in the cell, controlling all the activities of the cell. We can say the nucleus as the prominent organelle in the cell, and which is controlling all the activities of the cell. That is the main function of the cell. Now let us learn about the structure of the nucleus. As you can see here in the diagram, the structure of a nucleus. A nucleus has a nuclear membrane which covers its cell. See, nucleus has a nuclear membrane which covers its cell all around, and they are having pores. Nuclear membranes are having pores. What is the function of the pores? They will be allowing the movement of the substances in and out of the cell. So what are the functions of the pores that which is present over the cell? That the function of the pores are allowing the substances moving from one place to another, moving in and out. And also you can see here in the nucleus there are chromosomes. So rod-shaped structure present in the nucleus. What does it contain? It contains genetic informations. Then chromosomes contains mainly two things. What are they? DNA as well as proteins. What is the function of DNA? DNA is responsible and organizing for the constructing new cells. And proteins, we all know, the helping for the packaging. Condensation is taking place here. Condensation in the sense compaction for a gene regulation, we can say. And before that, before the condensation, the chromosomes will be found in the form of chromatin material. You can see here in the picture clearly. It will be found in the form of chromatin material, thread-like structure. So this will be organizing itself into chromosomes. When the cell is going to divide, it will condense. Let us see the condensed form. You can see here the structure of the chromatin material. First it is in the form of thread-like material. Then how does it see chromatin condensed chromosome structure? You can see when it is condensed, it forms in the form of a rod-shaped structure. The next thing that we are going to learn in the nucleus is about nucleolus. We can call the nucleolus as the brain of the nucleus. So we can call nucleolus as the brain of the nucleus. So it almost comprises 25% of the nucleus, and also it consists of proteins and RNA. What is RNA? Ribonucleic acids. What does it help for? Formation of the ribosomes, which is helping in the protein synthesizing. So it mainly formation of the ribosomes helping in the protein synthesizing. Then what is nucleoid? You can see here in the nucleus, nucleoid. So sometimes what happens, these cells will not be having a well-defined nucleus, and they will be lacking the nuclear membrane. So such nucleus we can call as nucleoid. When it lacks the nuclear membrane, we can call it as nucleoid. Hope it is clear to you. Next, we will learn the differences between the prokaryotes as well as the eukaryotes. You can see here the image of prokaryotes and eukaryotes. As you all know, the first difference difference between the prokaryotes and nucleus. You can see here there is no presence of nucleus. There is no presence of nucleus in prokaryotes. While in eukaryotes, the nucleus exists in the cells. You can see here in the image, nucleus exists in the cells. Here in the prokaryotes, the second difference is that it consists of a single chromosome, only one chromosome. While in eukaryotes, it will be of multiple chromosomes. Here in the prokaryotes, they undergo asexual reproduction. While in eukaryotes, sexual as well as sexual, both the way. The reproduction it undergoes sexual as well as asexual. And the next difference, main difference is that they are generally unicellular organisms. Here, while in eukaryotes, it is multicellular organisms. And here, as you see in the image, there are no membrane-bound cell organelles present here. While in eukaryotes, there are membrane-bound cell organelles. You can see here endoplasmic reticulum as well as the mitochondria, ribosomes, all the cell organelles you can find it in the eukaryotes. And the examples of prokaryotes are cyanobacteria, bacteria, blue-green algae, all these are examples of prokaryotes. And the examples of eukaryotes, you can say plants, fungi, animals, all these things comes under eukaryotes. So we ourselves have the examples of the multicellular. Shall I repeat the differences between the eukaryotes as well as the prokaryotes? Yeah, it's very easy, simple. Once again, I will just revise it. Here in the prokaryotes, there is no nucleus, while in eukaryotes, nucleus exists in the cells. And the second main difference is that a single chromosome is present in the prokaryotes, while multiple chromosomes are present in the eukaryotes. And the next one is that they undergo asexual reproduction, while in eukaryotes they undergo both ways, sexual as well as asexual reproduction. Here they are generally unicellular, while they are multicellular. And here there is no membrane-bound cell organelles, while in eukaryotes there are membrane-bound cell organelles. And examples we have learnt bacteria, cyanobacteria, blue-green algae. And in eukaryotes we have learnt the examples fungi, plants and animals. Hope it is clear to you. And the next prominent feature about the cell that which we are going to learn is cytoplasm. So we have come across plasma membrane and nucleus. Its function also we have learnt. Plasma membrane its function, nucleus and its function we have learnt. Now let us learn about cytoplasm. What is cytoplasm? The plasma membrane has a fluid-like substance in it, in the cell, that is termed as cytoplasm. The plasma membrane will be having a fluid-like substance that is termed as cytoplasm. And this cytoplasm will be containing several organelles. And what is the function of these several organelles? They will be performing distinct functions. What are the main functions of the cytoplasm? Let us discuss. That will be, it supports and suspends the cell organelles and molecules. The first main function is that it always supports and suspends the cell organelles and the molecules. And the second function is that formation of proteins takes place in the cytoplasm. And the next function is that it allows the movement in cell such as hormones and all. It allows the movement. And also the last important function is that it dissolves the cellular waste. Now let us discuss about the cell organelles. What are cell organelles? Its functions. Let us discuss about. What are the cell organelles? Let us see. Cell organelles are used for separation. That means it contains different functions. It will be attending different functions. Different functions will be performed by the cell with the help of this cell organelles. Important cell organelles are endoplasmic reticulum, Golgi apparatus, you can see here, lysosomes, mitochondria, plastids, vacuoles, centrioles, ribosomes, peroxisomes. All these are the cell organelles that which is present in the cell. Now let us learn each one in details. The first one that we are going to learn is endoplasmic reticulum. You can see here the image of endoplasmic reticulum. As you see here, it is a network of sheets like you can see here. So the endoplasmic it's quite similar to the plasma membrane. Network like structure you can see here. So it looks like a geese type, no? we do not know the ending or we do not know the starting, isn't it? So it consists of a membrane bound tubes and sheets. You can see here in the image it is clearly shown. So there are two types of endoplasmic reticulum. What are they? Rough endoplasmic reticulum as well as smooth endoplasmic reticulum. Then rough endoplasmic reticulum, as you can see here in the image, it contains ribosomes. What is the function of ribosomes? What is the responsibility of the ribosomes? Manufacturing of proteins inside the cell. That is the main function of the ribosomes. And the smooth endoplasmic reticulum, you can see here in the image here. It is a tube-like structure, you can see here. So what is the function of this one? Manufacturing of fats or lipids. That is the main function of smooth endoplasmic reticulum. So main differences between the two types of endoplasmic reticulum I said, smooth as well as rough. Rough, you will be having ribosomes over the body, over the cell, and in the smooth endoplasmic reticulum you can see here the manufacturing of fats as well as lipids. Then what are the functions of lipids and proteins? Let us see. So these proteins and lipids are synthesized on endoplasmic reticulum. Where is this proteins and lipids synthesized? On endoplasmic reticulum. For what? For making the cell membrane. This process is termed as membrane biogenesis. So this process is termed as membrane biogenesis. Now let us see the functions of the endoplasmic reticulum. What is the main function? As we have just now discussed, the transportation of material between the different parts of cytoplasm and between the nucleus, as from the cytoplasm to nucleus. So, and also another function of this one is holding of proteins, that is which is synthesized by the ribosomes. What is the main function of ribosome? As I have told you, it is the protein synthesizing. And another main function of endoplasmic reticulum is detoxifying the poisons and removing the drugs out of the cells. That is the main function of smooth endoplasmic reticulum. Detoxifying the poisons and removing the drugs out of the cells. That is the main function of the smooth endoplasmic reticulum. Now let us see the Golgi apparatus. Why it is termed as Golgi? It is discovered by Camillo Golgi. So it is named after the scientist. That is what the cell organelle is named Golgi apparatus. And as you see here, it contains vesicles and also it is arranged in the form of stacks. You can see here it is arranged in the form of stacks. And these stacks are called as cisterns, or we can call it as cisternae. And these vesicles will be having their own membranes, and these membranes are often connected to endoplasmic reticulum. And what is the main function of this Golgi apparatus? The main function is that it carries all the materials that which is synthesized in the endoplasmic reticulum to the different parts of the cell. And where is this material stored? This material is stored in the vesicles here, you can see here. And also another function of Golgi apparatus is that formation of complex sugars, and another function of Golgi apparatus is that formation of lysosomes. So now let us learn how to draw this endoplasmic reticulum as well as Golgi apparatus. Now let us learn how to draw the endoplasmic reticulum. It's very easy. How to just foldings you have to draw in this way. And the next folding will be starting from here. Ribosomes all over the endoplasmic reticulum, and these are the endoplasmic reticulum. And you can see here the smooth endoplasmic reticulum. Just you have to draw it in the folded form. Next we will learn how to draw the Golgi apparatus. Golgi apparatus. Okay, it is as I have told you, it is formed in the form of stacks, just bulging. So you have to draw it in this way, the Golgi apparatus in the plant cell organelles. And the secretory vesicles will be growing in this way, and this is the lumen here, this is the secretory vesicles here, and this is the Golgi apparatus that is as arranged in the form of stacks, and we can call it as cisternae. Hope it is very clear to you. So far what we have learned was, I hope that it is very clear to you how to draw it. It is very easy. So far what we have learned was the differences between the prokaryotes and the eukaryotes, and the three prominent features of the cell. What we have discussed was the plasma membrane, nucleus and cytoplasm. We have learned its function also. What is the main function of nucleus, the main function of cytoplasm, the main function of plasma membrane. We have learnt in details, and after that we have discussed regarding the cell organelle. What is cell organelle we have learnt, and only two cell organelles we have discussed today, that is endoplasmic reticulum, its function we have learnt, and also how to draw the endoplasmic reticulum in the cell we have learnt, as well as the next one what we have learned was Golgi apparatus, how it is formed in the form of stacks. Everything we have learned in detail. So hope it's easier for you. Do the revision properly and learn at home. Thank you.